Hello everyone and welcome to today's webinar, in vivo recording of spinal and DRG neurons to assess the activity of analgesic drug candidates, presented by Julien Allard. My name is Diane Royer. I'm the marketing manager at Neuroservices-Alliance and I will be moderating today's webinar. For those who already attended our webinars, this event is part of a webinar series that focuses on showcasing our members' innovative assays and technologies. Every webinar is presented by one of our neuroscientist experts. We know that your time is valuable. This is why our events all follow today's format, 20 minutes presentation, followed by 10-minute questions and answer. If you have any question at any time, please submit them in the question box that is located on the bottom of the GoToWebinar panel. I would like to inform you also that this session is recorded and you will receive the recording after the live session in a follow-up email. However, I strongly encourage you to stay until the end as it is the occasion to have today's speaker answer your questions live. Before starting, I will just say a few words about Neuroservices-Alliance. We are a consortium of CNS contract research organizations specialized in vitro and in vivo electrophysiological and behavioral studies. Our goal is to deliver endpoints facilitating decision-making to our clients for their CNS and pain drug discovery programs. We value direct and efficient interaction with our clients, and we rely on a team of more than 50 PhD scientists. And we are now ready to begin. Let me introduce you to Julien Allard that will present today's webinar. He's the founder and chief scientific officer of EFIS. Hello, Julien.
Hello, Diane.
Hello, thank you. And I will let you take the lead from here.
Okay, thank you. So thank you everyone for attending this webinar. So I will briefly present, in fact, some data and some approaches that we have developed at EFIS to test analgesic drug in vivo on electrophysiological endpoints. So a very brief reminder on ascending pain pathway first. At the peripheral level, the cell bodies of the nociceptors and of the mechanoreceptors are mixed together in the DRG. The axons of the mechanoreceptor enter the spinal cord through the dorsal horn. Some of them give off a branch, and the main axon is traveling to the brain ipsilaterally to form the so-called dorsal colon median lemniscus system. The axons of the nociceptors also enter the spinal cord through the dorsal horn, but there they make synaptic contact with so-called projection neurons. The axons of the projection neurons decussate and reach the brain contralaterally within the anterolateral system. The neurons that receive exclusive input from nociceptors are located in the very superficial layers of the spinal cord, lamina 1 and 2 outer, lamina 1 in the following presentation. There are other neurons that are involved in the generation of pain sensation that are located in deeper layer of the spinal cord that receive additional input from mechanoreceptors. So this is a textbook view, and it's only partially true because they are very clearly nociceptive specific neurons receiving exclusive nociceptive input in the deep layer of the spinal cord. In addition, some neurons in lamina 1 that are part of the anterolateral system do not participate to the generation of pain sensation. In rodents, the main ascending pain pathway is made by lamina 1 neurons projecting to the parabrachial area. This pathway is thought to play a major role in the affective component of pain, thanks to the projection of the parabrachial neurons towards the amygdala and the stria terminalis. The discriminative pain pathway that terminates in the thalamus have been much less studied in rodents. It is noteworthy that the receptive field of the parabrachial neurons that are involved in the generation of pain sensation is extremely large, for some of them encompassing the entire body. In comparison, the receptive field of the neurons involved in the discriminative pain pathway at the level of the thalamus is much smaller, encompassing only a very small portion of the body. Now, some technical considerations. So all the experiments we performed are done in anesthetized mice or rat, and the anesthetic of choice is isoflurane. We can use others. All experiments involve the catheterization of the jugular vein for systemic injection of any drug and required fluid, and the catheterization of the carotid artery for blood pressure measurement. In addition, the core body temperature is controlled and maintained at 37.5 degrees C. At completion of the experiments, we perform a complete blood biochemistry analysis, especially to ensure carbon dioxide and blood pH. To search for DRG neurons, we use electrical stimulation of the hindpaw while moving the electrode up and down in the DRG. We have two strategies to search for spinal cord neurons. When working with the wide dynamic range neurons located in lamina 3-5, the search is based on gentle mechanical stimulation of the hindpaw while moving the electrode up and down in the spinal cord. When recording from lamina 1 neurons, we use antidromic stimulation from the parabrachial area. That is electrical stimulations that will generate antidromic action potentials. And this ensures that the neurons we are recorded from do project to this brain region. So spinal cord neurons first. Those are examples of recordings that were obtained in anesthetized mice. Those 3 recordings comes from duly identified spinoparabrachial neurons using antidromic stimulations. At the level of the lamina 1, most neurons are nociceptive specific. That is, they respond predominantly to noxious mechanical stimulations or thermal stimulation. There are also a few modality-specific neurons at the level of the lamina 1. Some of them are so-called cold neurons that respond predominantly to water jet at 0 degrees C within our experiments. Deeper in the spinal cord are the so-called wide dynamic range neurons that, in addition to responding to noxious mechanical and thermal stimulations, also responds to non-noxious stimulations. So, as I said, there are also nociceptive specific neurons in lamina 3-5, but there are no wide dynamic range neurons, at least according to our experience, in lamina 1. So those are responses that were induced by natural stimulation, mechanical or thermal. It is also possible to record the response of spinal neuron to an electrical stimulation of the receptive field. The response then consists in 2 waves of action potentials. A first wave that is triggered by the recruitment of fast-conducting myelinated fiber, and a second wave that is triggered by the recruitment of slowly conducting unmyelinated fiber. If we assume that for those neurons, all the C fiber input is coming from nociceptor, this is a quite interesting feature because this is a pain-specific response. One thing which is important to keep in mind is that there can be marked rearrangements at the level of the spinal cord in pathophysiological conditions. And in the last decade, it's been shown that in chronic neuropathic pain state, there is the opening of a ventrodorsal circuit that allows mechanoreceptor that respond to light touch to activate lamina 1 nociceptive specific neurons that are normally only activated by noxious stimulations. And this is the, let's say, neural pathway for allodynia. It is possible to reproduce the states of so-called disinhibition by an acute application of bicurculine and strychnine on the spinal cord. And accordingly, we've shown that after application of bicurculine and strychnine, it was possible to obtain strong responses to light brush from neurons that were formerly unresponsive to light brush but only responsive to noxious mechanical stimulations. And this is quite important because it raises the possibility that in pathophysiological conditions, we can have a major dysfunction, a major allodynia or hyperalgesia without any change in the state of the DRG neurons themselves, just because of some forms of rearrangement at the level of the spinal cord. So I will now show you two examples of analgesic drug. The first is the well-known spinal morphine. So those experiments were performed in anesthetized mice, and we recorded here lamina 3/5 wide dynamic range neuron. This is the response to electrical stimulation of the receptive field. And as we said, we have here the C fiber related response, which can be considered a pain-specific response. The application of spinal morphine abolishes the C fiber related response within seconds. And while having still morphine on board, it is possible to completely reverse the effect of morphine with a systemic injection of naloxone. So, at least in my hands, but not only in my hands, in fact, systemic morphine is much less efficient on the activity of spinal cord neurons. In contrast, a drug which is extremely efficient on the activity of spinal neurons is Tetradotoxin. So we here again have measured the activity of lamina 3/5 neurons in anesthetized mice, and we have measured the response to noxious mechanical thermal stimulations and also noxious electrical stimulations. Here we measure the C fiber related action potentials. All those graphs show in fact the ratio of the response to baseline. And what it shows is that a systemic injection of TTX completely abolishes the responses to noxious mechanical, thermal, and electrical stimulation. And of course, these experiments were possible because we work in artificially ventilated anesthetized mice, because this dose of 20 microgram per kg would be lethal in a freely behaving conscious mice. And of course, the reason for this analgesic efficacy of TTX is certainly due in great part to the fact that it is an inhibitor of NaV1.7. Now, in some conditions, it's very important to use appropriate pathophysiological pain model to assess the efficacy of analgesic drug candidates. Here, we have used rats with a chronic constriction injury of the sciatic nerve to induce a state of neuropathic pain. We've measured the activity of lamina 1 spinoparabrachial neurons, and we've shown that in rats with CCI, there was a sensitization to noxious mechanical and thermal stimulations of the lamina 1 neurons compared to the sham rats. Note that we did not observe spontaneous activity in lamina 1 neurons in CCI rats, in contrast to what has been reported by some in the literature. So anecdotally, we've shown that acute pregabalin had no effect on the activity of the lamina 1 spinoparabrachial neurons in both sham and neuropathic rats. Now about DRG neurons. So we only, in fact, consider here unmyelated DRG neuron, and according to our experience, 99% of the recorded unmyelated DRG neurons, at least in our experimental setting, are nociceptors. It's extremely rare, in fact, to measure from a low threshold mechanical C fiber. So the response of DRG neuron is a bit different than the response of spinal cord neurons. The response to an electrical stimulation is one single action potential instead of many action potential at the level of the spinal cord. And the other difference is that many DRG neurons are modality specific. There are, for example, some cold-specific neurons that respond predominantly to water jet at 0 degrees C, and in fact, in a greater abundance, mechanospecific nociceptors that respond predominantly to pinch. There are also polymodal neurons that respond to both noxious mechanical and thermal stimulation. The best example I have to illustrate the distribution of the modality specificity of DRG neurons is coming from the study where we compare the activity of unmyelinated nociceptors in control mice and in mice treated with oxaliplatin. So the mice received here one single dose of oxaliplatin. The experiments were performed at day 3 and 4 after the injection. And I do not show it here, but on a behavioral endpoint, the mice treated with oxaliplatin showed mechanical allodynia and cold allodynia. So we did not find any difference in the activity of the unmyelinated nociceptors in vehicle and oxaliplatin treated mice. In fact, we recorded from 40 nociceptors from 16 vehicle mice and 50 nociceptors from 20 oxaliplatin treated mice. What is important is what is shown here. That is the fact that, in fact, more than half of the DRG neurons are modality specific. That is cold, heat, or mechano-specific. And about 2/3 are polymodal. The ratio of the response for those polymodal neurons, the ratio of the response to heat over the response to pinch, that is the number of actions potential generated by water jet at 50 degrees C over the number of action potentials generated by a pinch, range from 1 to 100%, which also clearly show that there is in fact a great heterogeneity within those polymodal neurons. So we do not have pharmacological data on DRG neurons at the moment, but we did test a protocol that could be used to assess the analgesic efficacy of drugs on the DRG neurons. And the protocol is very simple. So the idea is to search for an unmyelinated nociceptor by performing electrical stimulation of the NPO. Once we have a nociceptor, we characterize the responses to a wide range of mechanical and thermal stimulations. And this allows to choose the most appropriate noxious stimulation for this neuron. We then repeated the noxious stimulation 5 times every 10 minutes. And in between the noxious stimulation, we performed electrical stimulation of the receptive field. 23 mice were used, and it was eventually possible to record from one DRG neuron for 40 minutes in 19 mice. The first thing to note is that there was, for some neurons, a tendency for a decrease of the response to the noxious stimulations. And in other words, it seems that C nociceptors tends to desensitize over repeated stimulation. And this is something to keep in mind if we want to test drugs. In contrast, the latency of the electrically induced action potential, that is the conduction velocity, was extremely stable throughout the experiment. And the current intensity that was required to induce an action potential was also very stable throughout the experiment. And this is an illustration of the previously described protocol. The analog recording here shows the response to the 5 successive pinches, whereas the raster plot here shows the latency of the electrically induced action potential. And as you can see here, this latency is very stable. There is a major decrease in conduction velocity straight after the pinch, which is in fact in agreement with the literature and absolutely normal. And this shows here the response to one single electrical stimulation on an analog recording, with here the stimulus artifact and here the action potential of interest. So in conclusion, we can use in vivo electrophysiology to measure the analgesic efficacy of drug candidates on pain-specific endpoints for a wide variety of targets. And those targets include, but are not restricted to, mu-opioid receptor, TRPV1, and NaV1.7. And we can do those experiments in mice, wild type or genetically modified, or rats. And we can use animals in control or pathophysiological condition. And that's it for now, and I thank you for your attention.
Okay, thank you very much, Julien, for this very comprehensive presentation. Please, everyone, do not hesitate to continue asking questions. We do have like a couple of questions for now. First one, do you have any data in these models with specific NaV 1.7 channel blocker?
So yes, years ago, we did test the so-called compound 52 from Amgen, which was in fact, how to say that, officially released, and we bought it from a company, and it did show a marked analgesic efficacy on pinch and water jet at 50 degrees C in anesthetized mice, but I'm not sure of the selectivity of this compound, which is why I tend not to show the data that we obtained.
Okay, thanks. Now, what are the advantages of recording DRG neurons in vivo in mice when you have human iPSC-derived neurons or primary cultures from non-human primates available?
Well, I think that any in vitro work is designed to, in fact, accurately describe and measure drug-receptor interaction. But drug-receptor interaction does not tell you anything about in vivo efficacy. And this for, in fact, let's say, two major reasons. One is that once in vivo, the compound needs to get access to its target, which is very often not the case. And the other thing is that if we think about a DRG, it's an extremely complex structure with a distal end and a central end. And extremely important, there is a differential distribution of the different molecular targets along this along the structure, from the distal end to the central end. So we can have a very efficient compound against a target, but that eventually has absolutely no activity in vivo on the response of the DRG neuron.
Thank you very much, Julien. For now, we don't have any additional questions, so I think we can wrap up the webinar. So thank you very much for your presentation. And thank you very much, everyone, for attending. We will send you a follow-up email with a short survey that will open up in your browser just after the webinar. And you will also have access to the recording. And you will be able also to follow up with us and, of course, ask any additional question if you have any. Thank you very much. And thank you, Julien.
Thanks.